put these closer together for our last panel, but if it's in your eyes, we can also.
I think it's okay. I think I can just kind of cheat more this way. So,
yeah, that's fine.
That's really lit up.
Here,
Yeah.
and then, do you guys want some water?
I got some, thanks.
I could use a glass.
Okay.
So Cassandra, remind me where you are again.
I'm from Rhode Island.
Okay, Rhode Island. That's not intuitive, so.
Yeah. But I did an internship in Juneau for 10 weeks last summer.
Cool.
And did you see Cassie's poster?
Yeah.
She hasn't done. Okay.
We were just.
Okay, good. Okay, good. And you had that in the MSS as well, right?
Yes.
Yeah. That's where I like. As soon as I started, I was like, oh great, yes, I've seen this before.
That's cool.
So AMSS, nice.
Yeah, there were not nearly as many undergrads there.
At AMSS? Yeah.
This is Jeanette. She's the Fairbanks-based emergency preparedness.
Oh, cool.
I will go find Shannon.
Probably Ellen.
Yeah.
Since she's a popper. Oh, okay.
Used to have an MSS on it.
Oh, very nice. Yeah.
Chris, isn't she at Seattle?
Yeah. Well, it's Sinkatrap of Alaska.
Yeah.
Yeah.
I'm not.
Chris told me to look Chris Whitehead.
Yeah.
Maybe she's going there, or she's.
Right. Yeah. No, I went. I think well, maybe she was. I don't know. I've been to like 4 conferences since MSS, and so they're
Right, so she knows
I was just in Portland last week.
right.
For a marine mammal surrounding
Yeah.
coalition thing, which was very fun and cool, but did you hear about the big snowstorm there?
Yes. Yeah.
Oh, that was when I was there.
Oh, right.
Snow that night.
This is my second conference ever, so
Oh.
it's a bit less chaotic.
Yeah. Oh, these real needles. Just model over cells. Well, we do need it for the online.
Oh, are these being streamed as well?
Yeah, I think so.
Hi, so thanks everyone for coming to our panel on harmful algal blooms. I apologize I was late. I was very drawn into a student poster and I didn't follow the time. But thank you all for coming. This was a topic that emerged as the #1 zoonotic-ish disease in the CDC Alaska working group on zoonotic diseases. So it's a really important topic, and we've got some excellent people here to share different perspectives from their work. So I think we'll maybe start at that end of the table, and if you guys want to go ahead and give your introductions.
We're going to finish it with
Oh, we're going to finish with Kathy.
We can go to skip. Don't look.
That's right, yeah. Please. Okay, we'll start with Shannon, thanks.
Hello, my name is Shannon. I work for the Sitka Tribe of Alaska as the Environmental Lab Manager. Full disclosure, I'm from New York and have only lived in Alaska for a year and a half, but it's probably been one of the best places I've ever experienced in my life. So most of the work I do is with paralytic shellfish toxins and also looking at other toxins. And that definitely ties into food security. So although I'm in the laboratory, that's something I think that me and my lab team frequently are thinking about.
Hey, my name's Thomas Ferrugia. I work at the Alaska Ocean Observing System, and we do lots of different things at AUS, but one of them is we coordinate networks that focus on specific topics, and one of them is the Alaska Harmful Algal Bloom Network that I'm the coordinator of. And so I mostly want to talk about that and not so much about me. I don't do much in terms of actually monitoring or sampling or research. That's really everybody else up here. But this network was put in place to kind of provide a statewide platform for discussing monitoring needs and research needs and how to communicate about HABs and how to set up kind of efficient responses for harmful algal bloom events. And so really the heroes here are the sampling partners, which most but definitely not all of them are up there. They do a whole myriad of things to help us better understand harmful algal blooms. So you can look for the actual phytoplankton that causes that blooms and causes these issues with phytoplankton toes. Shannon's lab, they do a lot of toxin testing from those blooms in food items. There are like more advanced techniques we can use, and so I kind of put those things up there just to give you a sense of what it is on the ground to kind of do this harmful algal bloom work. But we are trying at the network, we're trying to kind of all have a space to communicate and talk about how best to mitigate this threat. That's a threat, the way I look at it, it's a threat both to food security in general and then obviously to food safety specifically, the safety of the food that you are consuming. And so we're trying to kind of all have this big kind of melting pot think tank in here so that we can better address those needs in Alaska.
Hi there, I'm Cassie DeBlois. I actually am an undergraduate student in Rhode Island, so I'm a bit far from home. But in the spring, I'll be graduating with a double major in marine biology and computer science with a concentration in math. The reason I'm involved with harmful algal blooms is because I got a scholarship from NOAA that allowed me to do research in Juneau, Alaska for 10 weeks. So it was my first time in Alaska, and yeah, I haven't been here long either. But I looked at specifically ESA listed Alaskan marine mammals, and I did a literature review on over 100 papers. And part of that literature review was looking at harmful algal blooms, how they've changed over the past 15 years or so, and how those effects have changed on marine mammals. I also looked at marine debris and diseases, but this talk is about harmful algal blooms, so I'll stay focused on that. Thank you.
Everyone can close their eyes. We're gonna scooch back a little bit.
Oh, it's way too many pictures of myself up there. Okay, wow. I'm excited to be here. My name's Kathy LaFay, and I work at Northwest Fisheries Science Center, which is NOAA in Seattle. And I am a research biologist there, and just to kind of give you a background on me, I have been basically doing the same thing for 20 years. I've been looking at the presence of algal toxins in marine mammals, and we have set up a program there. It's called Warren West Wildlife Algal Toxins Research and Response Network. So next slide. So we measure toxins in marine subsistence harvested and stranded marine mammals from as far north as the Arctic Ocean all the way down to Southern California. We've been doing that for about 15 years, tracking where these toxins are. I was involved in the very first documented case of HAB-caused marine mammal poisoning with domoic acid in California. If anyone knows about that, that's kind of where I started. I was a first-year graduate student. Nobody knew what was going on. These animals were coming in, having seizures on the beaches. And I happened to be studying domoic acid for other reasons. I said, I think it's domoic acid poisoning. Went to the Marine Mammal Center. If you know Frances Gullen, she handed me bags of organs and poop and stuff, and the poop was the jackpot. So anyway, the reason I'm telling you this is that I have a unique perspective in that I've been watching this for 20 years, and I am watching a northward expansion of domoic acid producing blooms, and then we also have proof of increases of saxitoxin producing blooms, so the PSP blooms, which are much more important in the Arctic now, and we can discuss more later, but domoic acid unfortunately is coming. So anyway, that's the network. Next slide. So most of you, it looks like you already know, this was just a food web diagram. Here we have a pseudonychia, which would produce domoic acid. Here we have Alexandrium, the vegetative cell, which would produce the PSP toxins. It's already kind of been mentioned. Warming ocean waters, increasing algal growth, not only for the vegetative stage, which is the normal bloom, but also for cyst germination. For the first time in hundreds of years, the bottom temperatures are warm enough for cyst germination. Cysts have been building up for the last several hundred years, so it's a new source. Anyway, next slide. So all of that together allowed us to get a massive EcoHab grant. And I just wanted to show you this slide because these are just some of the people that are involved. We have even more, but we're all looking at harmful algal blooms and their toxins in Arctic and subarctic food webs from the Beaufort, Chukchi, Bering Sea, and we have collaborators also in the Gulf of Alaska. And that's it. And I just, of course, I want to say that none of this would be possible without the native communities sharing their resources with us, wanting to know what the information is. We get a lot of information from subsistence harvested walruses and bowhead whales. We have about a 20-year data set with the bowhead whales. So is that enough?
Yeah. Yeah, I think that was really a great kind of quick overview, but if anybody has a specific question about what are HABs specifically, and we can definitely go into that. We just didn't want to do that if it wasn't necessary, but we're more than happy to kind of explain kind of the biology behind it. Maybe we can put up the food web thing up there, this one, just to show, make the point again, how much, well, we're still learning to what extent, but how many different species in our marine food webs we've found toxins in from harmful algal blooms. And it's really all of them. The ones that are the most immediately harmful are ones that are collected by humans from filter feeders that are filtering out those blooms that are toxic, because that's the most direct kind of link, and so you've got the highest levels directly going from animals into humans in that way. But we're also worried about what happens to the resources that we rely on. Are these levels of toxins in bowhead whales, in walrus, are they going to endanger the livelihoods of those resources and therefore the food security of the people that rely on them? So it's that kind of the two sides of that same coin, but just I think it's a good thing to kind of keep in mind that it's like two kind of separate issues, but they're very much related. So I think, sorry, I think we need actually to give the mic, I've heard, around so that online people can hear. Which is going to make it difficult. Right, Elizabeth?
Yes.
Yeah.
That's okay. I need exercise anyway.
Thanks.
Yeah.
2 questions. So you actually said it was defined as a zoonotic disease.
Yeah,
In
in the
what
CDC working group, it was identified as
Yeah.
the number one zoonotic disease threat. Although, and please, ask the panel about whether it's a zoonotic disease.
Yeah, I just wondered, how could you explain that it's a zoonotic disease? That was the first question, and the second was, so you're unsure about the effect on human health from the toxin in either the water or the sea animals, if they consume either.
I'll start.
Yeah.
Okay. First of all, zoonotic disease, I don't know. I mean, I saw that, I worked for NOAA, and that report came out, and it's like, the HAB stuff is number one on the, and I think they don't really have a good place to put it, and it's about marine mammal health, and it is a significant threat to marine mammals. So I think that's why it's there. Honestly, I think the pure definition of zoonotic disease, I don't know that makes really any sense in terms of a toxin.
Then I agree, because I'm doing research into zoonotic diseases for a while, and so yeah. So I was curious. I mean, I've recognized the threat from, from the blooming alga, but I didn't understand perfectly well how. But now I understand your explanation. They didn't know where to put it, and that makes sense.
Yeah.
Yes.
I don't know who's online, by the way. Okay, and then your question about do we know about human health impacts? We certainly know about human health impacts in terms of mechanism of toxicity. So saxitoxin is a sodium channel blocker. So basically it just stops nerve function. You can have all kinds of sort of symptoms up to the main thing, but the main thing would be paralysis of respiratory system. If you can, and you wouldn't be able to breathe then. If you can get on a ventilator, it's very interesting. You can be ventilated and stay there until the toxin finally lets go and depurates from the body and recover. And there's been just some horrifying story of a person being poisoned and then being in the hospital completely not able to do anything, and that person hearing the people talking around like, should we pull the plug? We don't know, we don't know what's going on, so it'd be a really terrifying experience. And then when the toxin was finally out, he was fine. For marine mammal health and wildlife health, obviously there's nothing that can be done because you don't have hospitals for them. But so anyway, so talking with public health and hospitals, et cetera, is important in the community as this risk increases. But the main route of risk for human exposure in the mainstream world, not so much subsistence, is clams. So it causes paralytic shellfish poisoning. There's a lot of regulations in place. There's a regulatory limit of 80 micrograms per 100 grams shellfish. That's got a big safety factor involved in that. But there's not much known about some of the other vectors that people might be eating. And some questions that have come up from the communities have been like tunicates and anemones and things like that. We know there are many fish species that will accumulate some, but honestly the most is really has been in the shellfish where the risk has been.
Very interesting. Thank you. Thank you so much.
I think part of that too is that when shellfish are consumed, oftentimes the entire shellfish is consumed. Maybe not so much for scallops, but for everything else, where toxins have been seen to accumulate more so in the GI system as it's passing through. So if you're eating something and you're not consuming that portion, the risk of high consumption is lowered. So the amount of saxitoxin for paralytic shellfish toxins in particular present in the organism overall is not consistent throughout. So the way in which we're ingesting these things is also going to influence how we are at risk for these diseases.
Yeah, I think that is always something good to think about, is that we know well how it's impacting humans when we eat like shellfish directly. We don't know how it's impacting animals and at what levels those impacts start happening. But I think Kathy's got a little paper coming out soon about what organs these toxins are coming up in walrus and in bowhead whales. And so, are we eating those organs, or are we eating other organs where the accumulation isn't as great? So that's something to say. But like Shannon said, we usually eat the whole clams, small fish, sometimes we eat the whole fish. But for fish as well, there is a division as to where those toxins go, and usually they go in the kidneys, the liver, those kinds of organs. And so the muscles, and crabs, same thing. The hepatopancreas, the crab butter, that accumulates a lot of toxin, but the muscle doesn't so much. So, there are like nuances there where some threats could be mitigated by eating only certain parts of the organisms. I just want to make one quick note about the zoonotic disease thing. So that came out of a workshop that informed this report, and the workshop was focused on zoonotic diseases, and it's the people organically in the workshop, from what I understand, I wasn't there, organically in the workshop that said, well, what about HABs and PSP? We should include it in this because that is a disease, and quote unquote, that is coming from us interacting with animals. And so that's where it came from. But if you look at the report, there's actually an asterisk next to PSP, and it says like, this is not officially a zoonotic disease or something like that. But it was really is because people were they were there to discuss what is a threat to them in the natural environment coming from animals, and this was one of them. So that's the background that I heard about that.
Sure. Also, another thing with impacts on humans is we know that some of these organisms can have the toxins within them, but we don't necessarily know how fast they can eliminate those toxins. So part of the problem with shellfish is they accumulate things so quickly and they can't get rid of it very quickly. Whereas something like I read in one paper, the squid, they can accumulate these toxins, but they can actually eliminate them quite quickly. So we're still not sure at what levels, whether it's fish or marine mammals, how fast, we might know how fast they're accumulating based on environmental conditions, but we also don't know how fast they're getting rid of them and how quickly that might affect us down the line.
Sorry, we'll get to another question.
Yeah.
For sure.
I was just going to speak to the point that there is a lot we still don't know, and that's like an exciting prospect when we think of it from a scientific perspective, and a scary prospect when we think of it from a public health and food security perspective. But even about depreation rates. So Sitka Tribe of Alaska works with cedar partners throughout Southeast Alaska, and we get shellfish, and we had a sample from cloac of blue mussels that were at 1,600, and the regulatory limit is 80. 2 weeks later, they were below the regulatory limit. So blue mussels are really quick filter feeders, and they are coined our sentinel species, right? We'll see spikes in these toxins and know that the water has been affected pretty quickly. Butterclams can hold on to it for over 2 years. Little necks and cockles, even when we see these blooms, tend to be low. And I can't statistically speak to that. I can speak to it from just having tested the areas, and I never did an actual analysis, but you'll get blue mussels that are really high, butterclams that are high, and little necks and cockles that are higher than we normally see, because there's usually below detection limit, and maybe they're at 15 or 20, which doesn't mean that they're always safe to eat, because we have seen them over as well. But it is interesting to try and continue to learn about why we see these differences.
My question is, it's a little bit different from what we were talking about just there, so you'll have to forgive me. My name's Maureen Clinton. I work on fish, and I know very little about marine mammals. But I'm interested to know, I know invertebrates, saxitoxin, as you mentioned already, is a kind of neurotoxin, and it can impact neurological symptoms in those animals. And you have this fantastic large data set on bowhead whales, for example, and you mentioned that a lot of that data comes from strandings, but also subsistence fishers. I was just wondering if you could speak to whether you think that saxitoxin is a factor in some of those strandings. Do you see a difference in toxin loads between those two groups overall?
Hello. Okay. Great question. I'm going to give you a ton of information. You do fish. So the thing about saxitoxin is it's a fundamental sodium channel blocker. So it really impacts anything with sodium channels, except for how incredibly cool it is that there are clams that have a single point mutation that makes them tolerant to saxitoxin. You can see that genetic dynamic from low exposure regions where they don't have it, changing halfway up and up until they all have it in places where there have been PSP for multiple years. So that's that. Fish. Fish are also sensitive to saxitoxin. I've personally done studies with larval herring in dissolved saxitoxin at levels that would be ecologically relevant in a dense bloom. This is dissolved, not eating, taken through the gills, and causes paralysis, which then they recover from. So there's just a lot of information about mechanism. Marine mammals, bowhead whales. So of the 20 years of bowhead data that we have, it's hard to, this is something that we have all talked about multiple times, it's really hard to understand what a concentration you measure in the feces of a marine mammal is as to whether it was high enough to cause symptoms. You don't know how much they ate, you don't know when they got rid of it, you don't know how long it's been there, etc. So I've been just setting a scale of saying, let's think about this in relation to the regulatory limit for humans. So if it's at near the regulatory limit for humans in feces, you might say they're eating dangerous prey, at least close to dangerous levels for humans. We'd expect larger animals to need less of a dose to have impacts compared to smaller animals. That's a known toxicological phenomenon. And so when you think about that, the bowhead whales that we've been looking at, and these are subsistence harvested. They're not stranded, they're not ill, so they're presumably fine, unless maybe they are impacted, they're easier to hunt. But those levels have not been near regulatory limits, but they do fluctuate over time. Same with the other toxin, domoic acid, which we're not talking about here. Domoic acid is there. It's in this. It's throughout all the same regions, the same places, but it is not near as high in terms of regulatory limit. And so that's something to be thinking about. Saxitoxin is a very potent toxin, 80 micrograms per 100 gram. Domoic acid is not really super potent. It's an excitotoxin, overstimulates, but it has to be 20,000 to be, yes, 20 parts per million. Anyway, so in terms of, do I think that there are harmful algal bloom toxin impacts in marine mammals in the Arctic? Yes, I do think there's the potential there. Walrus populations, we now have 4 years of subsistence harvested animals sampled. We see 100% of walruses in the warmest year had toxin, and 2 of those had levels near the regulatory limit. That, to me, in terms of all the thousands of samples we looked at, is a definite warning sign that there are likely times when walruses are getting doses high enough to cause health impacts. But we're continuing to look at that to see, and we've had a report or 2 of a strange behavior, but we have that kind of out there too. Tell us what you see when you see an animal. Does it look like it has some sort of health impact? So.
Could you maybe just explain the regulatory limit and what that level indicates and where that number comes from?
Okay, so yes. So don't be confused with the real numbers, but just think about this in the abstract. I'm going to tell you, so if we measure a sample and we can say there's this much toxin per gram. So if we take out that silly 100-gram thing, the regulatory limit, if you're measuring toxin in something, is really 800 micrograms per gram for saxitoxin. If your seafood, whatever it is, even if it's a fish and most of it's in the gut, you're doing the whole animal, anything that's 800 micrograms per gram is considered unsafe to eat. And that has like a 10-fold safety factor in there too. I mean, that's based on, someone did a study, how much do people usually eat in one meal, and then from the real poisoning events with humans, what they predicted they had eaten. For domoic acid, that number would be 20,000 micrograms per gram. So that gives you an idea. You have to have that much more toxin. And again, that's a 10-fold safety factor. And that's eating a meal of 5 large razor clams in one sitting. And we've done a lot of work with mouse models, exposure models, and zebrafish exposure models to look at repetitive low-level exposure, which I don't know that we want to bring up here, but that there are effects with repetitive low-level exposure below.
That was going to be my next question.
So we do want to go there.
Okay, well, let's hear a question.
Yeah, so I was just wondering if there's research being done on repetitive low-level exposure for humans.
So I'm going to overlap with the HAB toxins. We've done most of our work in domoic acid. That's how I started. That was that first event in California. Saxitoxin, there have not been a lot of chronic low-level exposures with that. But I'm going to give you an example with the domoic acid. So acute domoic acid poisoning is called amnesic shellfish poisoning, causes seizures, overexcitation of nerves, permanent memory loss through lesions in the brain. they then find if an animal doesn't die, that it continues to have something called a chronic condition, and you might see these sea lions on police cars downtown or etc. And this person, Peter Cook, did a phenomenal study with sea lions in a maze where they had to make a choice, and then they took them to the hospital and did MRIs in the brain, and they found that the hippocampal lesion size that was caused by domoic acid poisoning was correlated directly with their inability to learn, spatial learning. So that's one kind of chronic impact, right? That was an acute exposure. We did studies where we looked at weekly low-level exposure in mice at doses below any signs of impact. So the mice are just acting completely normally, but exposed once a week for 25 weeks. And then we did tests in a maze, so not as cool as the sea lion, but it's a little radial maze. And we found a significant depression in spatial learning. They could not learn how to navigate the maze, even though they looked completely healthy and they had none of the classic signs of amnesic shellfish poisoning. We stopped the exposure. 9 weeks later, tested them again, and they were back to normal. So it was a reversible impact, which is good news. But again, this became much an interest in the tribal communities of the Pacific Northwest that are known to be regularly consuming low levels of toxin. So that's been sort of an ongoing thing that we've been looking at. In terms of saxitoxin, the mechanism is so basic. I don't know how to predict what chronic low-level exposure to saxitoxin would do, because it's not as pinpoint of a neuro impact like the domoic acid, which is impacting the glutamate receptors. But there could certainly be some sort of impairment in behavior or abilities with chronic low-level saxitoxin exposure, but that is not as well known.
Thanks. Question. So there are a number of different congeners of saxitoxin, and so the question is, is the toxicity of all of them equal? That's part A. Part B is, do you know what the metabolic pathway is, such that if you give an animal, or if an animal receives saxitoxin, is it really GTX4 that is the problem? And how many steps does it take to get to GTX4 and that kind of thing? What do we know about metabolism that might help us in the long run either try to speed up the metabolic process such that you don't have to wait on the ventilator till whatever lets go of whatever, but you could speed up the metabolic process such that you get through that GTX4 faster. Can you talk to that a little bit? I think I'm directing it at you, Kathy.
The second part. Do you want to answer the first part about congeners? Yeah, so all the congeners have different toxicity factors, and that when we were testing on HPLC for toxin level, it's a multiplicity factor, so you're multiplying it in, where saxitoxin has a factor of 1, and then all the other congeners are assigned relative toxicity factors. So your C toxins tend to be less toxic. One of the congeners that I can't remember which one is like 0.01, and again saxitoxin is 1. So if you had a lot of that congener, right, we would assume that we wouldn't feel the effects of toxicity in the same way we would with the same level of a higher toxicity congener being present. And I think there is a lot of interesting research that is happening, as we saw on the poster in this poster session, but that can continue to happen about congener presence, because that will be really, really important for identifying where this toxicity is coming from if we're looking for better rehabilitation methods.
Sorry, Shannon, can you define congener for molecular biologists?
Yeah, well, I can try. So a congener, going back to chemistry, and I'll try and make it not too confusing, too chemical, but if we think about a molecule, they have functional groups, which is just different parts of the molecule, and oftentimes you'll have a suite of molecules or molecules that have the same basically base form with a different functional group. So a sulfur or a nitrogen where something else might have been, but they can still function in a similar capacity. So these congeners have different functional groups that affect the way that they're binding with our sodium ion channel in terms of strength and basically length of time bound to those. But they still act in that same way of impeding that sodium ion channel. So it's just a grouping of toxins that are molecularly a little bit different, so they behave just a little bit differently.
And in terms of that, so that suite of toxins that we're talking about, Alexandrium, so let's start at the bottom, the phytoplankton, Alexandrium is going to produce a suite of toxins, that suite of toxins that we were just talking about, saxitoxin being one of them. Different strains of Alexandrium in different parts of the world, or etc., produce different amounts of each of those toxins. So some strains would be considered more toxic because they have more saxitoxin, etc. So we've been looking at that. This is part of that EcoHab project. This again, Don Anderson's lab at Hui, and Evie Fashon has been doing a lot of this work. And we're finding that in the Arctic, this is Beaufort, Chukchi, North Bering Sea-ish, most of the phytoplankton samples that we've gotten are producing a very similar suite of toxins. That's very convenient. That's nice for our models, right? So a certain, and I can't tell you right now, but we've recently published a paper that shows in a pie chart each concentrate or the amount of each type of toxin that this phytoplankton produce. Okay, so that's there. Then you have the clams. So the clams are filter feeding, and they're feeding that on that suite of toxins, and then they metabolize it. And after certain periods of time, they could make things more toxic. They could make things less toxic. So we have a suite of toxins in clams. Again, these are snapshots, though. This. We started to see a really cool thing where we could see that the clams looked like they were in various stages of biotransformation, because they finally got to a certain place, but you could see, based on what we know about which toxins they do first. So anyway, so that's interesting. Okay, so we have suite of toxins in the phytoplankton, some sort of toxicity level. Suite of toxins in the food you're eating. So I'm going to go to walruses because that's what we're interested in. Everybody should be interested in. And then the walruses are going to eat these clams, and it's a suite of toxin. They have the potential, the pH in the gut, etc., to have some of that happen too. Go to more toxic, go to less toxic. So we are working on all of that. We're going to get the suite of toxins in the feces and in all the organs if we do find toxin levels there. So we'll have a really good story there on what that means. But your really cool question about treatment, can we push this along to make it happen? That is, I don't know how we would do that. Once we understand how they're biotransformed, smarter people than me could look at that, and maybe there is something. I'm just trying to imagine how that would even work, though, because it's going to be sort of enzymatic processes.
Right, but it could work through something as simple as pH, right? If you see that what goes in from the clams go into the walrus, and then you see the walrus gut content is radically different than what went in, we already know that presumably something in the gut has accelerated the biosynthesis or the metabolism of the different compounds. So, I mean, it might be that something as simple as acidifying it would help speed up the process of metabolizing those compounds.
So I know okay, that part I get, but you mean like how, after the person has consumed it,
Yeah.
changing the acidity in the gut.
Yeah.
Hmm?
I mean, it could be
Could
something
be.
simple, but
Yeah,
it
yeah.
may
I like
be
it.
far more complex than that.
I think if you're in a place where you can acidify someone's gut, you probably have a ventilator nearby, like you're probably already in a hospital, and that might be, because I'm sure there's a lot of other things that happen if you try to acidify, but it is really cool. It is a really cool idea that I never thought about, so thanks for that. 2 quick points. One is USGS also does a lot of work. US Geological Survey does a lot of work on this as well, especially on birds, and there's a couple of researchers there that have been looking at how congeners have changed after they've ingested. So they actually dosed common mers in captivity with a known congener profile, and then seen how that changed when it was in the MERS. So there's definitely a lot of research being done on that thing, and that's really interesting. But the larger point I wanted to make is, so we've been talking for like 30 minutes. Look at the number of complexities in this problem, and it really goes to how difficult it's going to be to really get like actionable things done other than keeping people from eating something that's toxic, because we got to know when this is blooming. Okay, that's a whole set of forecasts with environmental parameters. Then when is it getting toxic? There's evidence that they're not always toxic. Sometimes they turn up that gene that makes them toxic, sometimes they don't. Then how does it move through the food web? Then how do those different congeners with different toxicities change in proportion in each of these individuals? I mean, that's a lot to take in and to try to track. And so one thing that I just wanted to point, after this little bit that we talked about is, we will probably never, not in the near future, get to a place where we can say that one specific, that to predict, to forecast whether something is going to be toxic or not, because of this level of complexity. So it's going to be a lot about communicating, outreach, being able to, if someone's feeling symptoms, know what they're feeling, like why that happened to them, and then communicating that to the rest of the people around them, that hey, it looks like there's PSP in the water, we should probably reduce our harvesting activities around here for the near time. So the work that CTOR is doing, or the Sitka Lab is doing, is really great in doing that, because they have that kind of local network of information gathering and then redistribution to the communities. And the way I see it is that with all this complexity, that's really the near term solution that we have is in that realm rather than in the prevention realm, which I think is going to be extremely difficult due to all these complexities that we've been talking about. So I just want to kind of put my opinion on that, to kind of bring it all back to kind of what can we do with all this information.
Kathy, you mentioned in your introduction, you just mentioned in your introduction that you think that pseudonychia and domoic acid is, I think you said it's coming. Can you expand on that? Yeah.
Yeah, so in 1998, and I heard you mention this, but I was a first-year graduate student at UC Santa Cruz, and there was this event over Memorial Day weekend, and we talked about that, where these animals were coming in, and that was the first documented poisoning event with domoic acid in marine mammals that had been ever documented in Monterey Bay area. And this was, this is a big mystery, and we figured it out. It was really great. And then it's been happening every year since. And when you look at marine mammal-centered data prior to that, they could start to see, oh, there was a few things maybe a little earlier. So I took that data, and when you look at the earliest record of a potential DA toxicosis in a marine mammal, or sea lion, in this feeding on anchovies, that was my thesis work. I was looking at anchovies. To now, there is a statistically significant increase in the number of DA toxicosis cases in California sea lions through to now. Dozens to hundreds of animals a year. So I was there at ground zero, right? So I've been looking at this. We had this Warren West program. And then in 2015, you remember the blob? The big warm water, the blob came in. We had the largest, longest lasting, most toxic pseudonychia bloom on record on the West Coast. And that was all along the West Coast. And I got a call from Department of Health saying, we have a seizing sea lion on Washington coast. So I had never seen a seizing sea lion for a decade, etc., until that event. They sent me some feces. That's what I do. Get lots of feces from all over, and it was high in toxin, and there was a seizure. So that was it. So there was this sort of indication that there's this that there can be a northern expansion of these phytoplankton. Now, the story with temperature is much clearer, and with Alexandrium. There's good evidence that we have other studies. The jury's still out on temperature with pseudonychia. I'm just telling you what I've experienced. So, in this Warren West program, we also have looked at ice seals, the 4 species of ice seals, bearded seal being one that eats benthic. The prevalence of domoic acid in bearded seals, this is not concentration, this is just how many animals have it. We plotted that for the 10-year period. There's a statistically significantly higher probability of having the toxin as you move through time in the North Bering Sea for bearded seals. Those levels are low. Those are not, I don't think that those toxin levels are high enough to be causing health impacts in bearded seals right now. But as a biosampler, it's basically sampling the environment, we're seeing the prevalence or the probability of having the toxin present there has increased. So that is just something I'm thinking about as we're moving forward and as these things go. It does appear to me that there is a northward expansion, that conditions are becoming more favorable for Pseudonychia blooms. We see Pseudonychia, we see domoic acid and saxitoxin, the Alexandria blooms, together all the time.
We do see these species in our phytoplankton toads throughout Southeast Alaska. So Pseudonychia is present, but as I hope Thomas will expand on, toxin does not always ensue with presence.
Yeah, and I'm not an expert. I just know kind of what I read in articles, but it does seem that at least for Alexandrium, there seems to be, and a lot of this comes from lab studies, there does seem to be an upregulation of the genes that end up by producing the toxins when you have a presence of a copepod or filtrate from a copepod. So a couple lab studies have shown that. And it makes sense. I mean, it's a defense mechanism. Some people have suggested it's a defense mechanism so that the feeding rate of their predators goes down, because they basically slightly paralyze their predators when they're toxic. And so, the idea is if they bloom but they don't have any predators around, they're not going to put in the energy in producing that toxin needlessly. So in certain, so for Alexandrium, for example, we know that there's obviously Alexandrium blooms up here, and we know that a lot of them are toxic, maybe not all of them, but a lot of them are. For Pseudonychia, we know that's around a lot, but we haven't detected domoic acid very often in clams from what I hear, at least not very high levels, and same with marine mammals. And there's a third one that we're kind of monitoring because it's created problems in British Columbia. The phytoplankton is called Dinophysis, and it produces a toxin called okadaic acid. And that one, we do see Dinophysis, but there's no confirmed record of okadaic acid poisoning or even any really. I think your poster shows a little bit of okadaic acid in some one case, yeah. Right, out of 200, yeah. So very little, and it may just be that either they're at the northern edge of their range, and so they can't produce the toxins because of environmental conditions, or it could be that they're not detecting the specific predator that they would need to detect in order to turn on those genes. That's my understanding of it, but it is probably rudimentary.
Something I also wanted to mention is outside of these toxins, harmful algal blooms can be harmful outside of being toxic. Like if the blooms are too big, and like the big chaining algae can actually block fish gills and things like that. But also if there's way too much of them, they can block out sunlight, and then when they all die, they'll take all of the oxygen out of the water and have those sorts of effects too. So we can have anoxic conditions due to harmful algal blooms, and that can be entirely unrelated to the toxins. That can just be because there's too many of them, because there's too many nutrients in the water and it got warm and there's just a lot of algae around. So although we're talking about specifically toxins here, there also are a lot of species that can bloom that don't produce toxins, or even the species within these genuses we've been talking about, or if they just don't produce toxins during that bloom, they can still have harmful effects.
That is a very good point, yes. And we should have probably started with that, with like all the harmful effects and then some of them being toxic, yeah.
Random, I don't know why this popped in my head. Is there any information about these about the blooms in relation to nutrients downstream of agriculture runoff?
So I'm not as certain with the Alexandrium, but there have been a lot of studies for Pseudonychia. The one of the things that's related is the runoff from agriculture, the pig farms in Monterey Bay, etc. So definitely nutrients play a role in those bloom initiation stages. When you're talking about toxin production, that work by Hans Dahm is pretty incredible. That's with the copepod feeding studies, but it's not consistent.
Right.
But anyway, so toxin production, for example, in Pseudonychia, if you are limited in one nutrient, that sometimes increases toxin concentration. So there's been a lot of laboratory studies that are showing certainly nutrients play a role, not only in the size of the bloom, but in the toxicity of the bloom. And you have to have enough of the right nutrients, but you also, if you have a limiting nutrient, you might get more toxin production.
I'd just like to make it a little one world, one health real quick, but a lot of these really rural communities in Southeast Alaska aren't having agriculture, or there's so much. You'll go out on a boat and you'll be like, oh, that's town. Like, you need the binoculars because you're surrounded by so much, but we're still feeling climate effects that are affecting this food security and these blooms and these resources. So, agricultural runoff is definitely adding nutrients and things, but it's just, I think it's important when we're asking questions as scientists or as community members and humans to remember that the world is so interconnected, and even these rural communities are going to feel the effects of warming waters and bigger blooms or more nutrients upstream.
And also, I think there's also evidence that even if there's no nutrients, like glacial runoff, like freshwater input, can also influence Alexandrium blooming, and areas of actually lower salinity, like estuaries or lagoons and stuff like that, maybe conducive to blooming of Alexandrium as well. In the lower 48, obviously, you have big problems with harmful algal blooms at point sources of high nutrient input in the Gulf of Mexico and stuff like that. And the only good thing about that is that there's a human action that you can take. You can try to reduce that runoff to help the harmful algal bloom. Here in Alaska, because we don't have that problem, we don't really have that lever to pull. But our limitation, probably more often than nutrients, is temperature and growing period, and so that's why there's some concern up in the Arctic with as the climate changes. We not only have warmer waters, which is more conducive for the germination of Alexandrium on a shorter time scale, but we also have a longer ice-free period, and therefore longer periods with sunlight, and therefore a longer growing season there. And so I think the limitation is mostly with sunlight and temperature up in Alaska, and that's probably not going to get better in the future, unfortunately.
I was curious about the response from commercial fishermen to these algal bloom events. What's their course of action when they see toxins in the water?
Yeah. So the problem is that they're not going to see the toxins in the water. In fact, to the naked eye, it's hard to even see an Alexandrium bloom in a lot of cases unless it's extremely dense. For commercial fishermen, I think the place of biggest concern is with crabs and gooey ducks, not so much for finfish. And then mariculture is the other kind of commercial aspect that is very interested in HABs and how that can affect their commercial harvests and selling their commercial product. And so in those cases, I think there's more and more of them that are interested in being part of monitoring, so that they kind of know whether or not they should go out and harvest their oysters out of their farm, or if they can somehow avoid times when it's more likely that the product is going to be full of toxins. And then on the commercial side, for all the mariculture firms, there is a DEC it's a regulatory requirement for all of them to send in samples before they harvest and sell their clams out of their farms, that they get tested for PSTs. And if it's too hot, they get shut down for until they have multiple tests in a row that are below that regulatory limit. So the commercial side is actually very well regulated in that sense, but that's a very big contrast to the subsistence food harvest side, which has no requirements and no infrastructure really to regulate that. So it's, yeah.
That's great. The commercial part is good. I will mention that I work for NOAA, and so this large collaborative project that I was talking about also has the Alaska Fisheries Science Center and all the folks that answer to the Pacific, the Management Council for Fisheries, and they want to get an indicator. So we have a meeting once a year. What is the potential for HABs to influence fish stocks? So there is interest in finfish, but again, finfish, the commercially available finfish, have not historically had concerning levels of toxin. It's there a little bit, but they're wanting to put that on the radar, and they're wanting to say, hey, can we get some sort of indicator, like it does temperature and ice cover, can we correlate to say what's the risk for this year? And that is getting at some of the work that we're trying to do in the EcoHab project, is model under what environmental conditions do you expect there to be higher blooms, under what bloom conditions, what cell counts do you expect there to be dangerous levels in the food web. We are going to be able to model that. We have 3 years of data right now with different environmental, it's just a matter of doing it. We're working on the biological part right now, bloom density, how much you're going to see in the prey. You're eating this diet, the walrus is eating this array of things, how much are they going to get in a day, in a week, etc. So we can start to make some of those predictions. But Thomas is totally right. Globally, the regulations for testing shellfish is really solid.
It also makes me think of halibut and rockfish and other benthic fish. Are they like more likely to have toxin than like the deep water fish?
Yeah, we find this interesting scenario where filter feeding fish like herring, anchovies, those guys they're just going through the water, and they're just filtering all this, and they're getting all this toxin. They can be quite toxic, and then there are fish kills, herring fish kills in the past from that. The nice thing about domoic acid the nice thing about saxitoxin is, for those fish, when they do get high enough, they will die. So there'll be a fish die-off. For domoic acid, they seem to be very tolerant. They can go around and just stay in the water column and feed all the marine mammals this high dose. But we see this lag in the benthos. You'll see higher toxicity in the pelagic area first at the beginning of a bloom, and then kind of this delay, and then you do see it at the bottom. The benthic species halibut, you have to think about what they're eating, and Shannon talked about how if you're not like filter feeding like a basket, like a clam or an anchovy, you're not going to get as much toxin per unit body. It's mostly going to be in the gut. So, I don't want to say things are safe. They need to be checked out, but those kinds of fish are not going to be as big a concern unless you were just eating a bunch of GI tracts. Right.
What do you think it would take to get the subsistence systems more in line? Like, yes, we have this issue with subsistence monitoring of these toxicities. What kind of policies would you envision to make more resources available to the rural communities?
So you guys do the Gulf, and then I'll talk about the different issue about.
Oh yeah.
Yeah, so SICA Tribe of Alaska has worked, and it's now its own consortia, but the Southeast Alaska Tribal Ocean Research Consortia is 17 tribes throughout Southeast Alaska working to monitor their resources. So currently, this is, I hate to use the word grassroots because it's government, right? It's government, tribal governments, but sometimes the authority that they are prescribed feels more like it's a grassroots issue. but these governments are monitoring their resources because Alaska is the only state that doesn't do coastal monitoring the same way that the lower 48 does. So we have the most coastline. We have, I can't say in specific numbers, but per capita, I would imagine the most coastal communities, and especially rural coastal communities who will struggle to get access to food as well as healthcare for these toxin-causing diseases. But that's its own issue. So these tribes have come together to monitor their beaches, and Sitka tribe tests. We have an environmental laboratory where we are testing for saxitoxin, or I should say the paralytic shellfish toxins, because the assay we run tests for all of those congeners that can cause toxicity. And we're also starting some baseline monitoring for okadaic acid as well as domoic acid, which are for diuretic shellfish poisoning and amnesic shellfish poisoning, which is what we're talking about with the sea lions. And this really provides the tribes the ability to monitor their resources, which they have not been given access to previously. The state does do testing, which all commercial needs to be tested by the state. However, it's expensive, it's a lengthy time wait, and for individuals who are just trying to put food on their table, that is not a practical or viable solution. So again, CEDAR has been sending us samples. All of our partners ship throughout Southeast Alaska, and we test the samples. And we are a non-regulatory body, but we go by regulatory guidelines. So if there is shellfish that is over 80 micrograms, we are going to release a public service announcement, and we also release all the data so people can see the toxin reports. You can all sign up for our listserv if you're interested, but there will be a PSA saying this beach was high in toxin for this species, and if it's blue mussels, we advise no harvesting at all, because again, those are the very quick filter feeders, so toxin levels are probably really high in the water, even if maybe not all of the species are representing that right away. I think this autonomy is great. However, it's unfortunate that it has fallen on tribal governments to handle, because if you are receiving funds through IGAP, and that is your only source of funding for all of your environmental monitoring, PSTs is a fraction of the things you have to deal with. You also have to deal with waste disposal. You have to deal with other food accessibility issues, other things I don't think about because I work in the lab that tests for toxins. So that's all I think about. So I think the solution is giving more sovereignty to these governments to monitor their resources, and that happens in the way of funds and access.
Yeah, thanks for the question, and that was fantastic, Shannon. I think the only thing I'll add to it is that unfortunately there's no, because of the congeners we've been talking about, there's no easy test. You have to test in the lab, essentially, and even some of the tests in the lab won't give you the full suite of congeners. It'll maybe give you an indication of the toxicity, but not the full toxicity. So if you use one of those tests and you're at 40, you're below 80, what do? Do you try to test it more? And all that takes time. And when you're trying to talk about someone's meal, like you were saying, deciding whether or not to feed to their family, you can't wait days and days. And that time has to include you shipping the sample, making it out that day from wherever, King Cove, getting to a lab, in Anchorage, for example, having it tested, and then those results going back to you. I mean, that's at best that's 3 days. So you could have more labs, but then you need space for those labs, and you need staffing for those labs. And where do you get those? Everybody in these communities are already doing 15, 20 things, right? Now you're giving a job to somebody that they have to add to all the other duties that they have. But it's not probably a full-time job, right? Because this only happens every once in a while. People go and collect at low tide only, during the lowest low tides or whatever. So you have to kind of meld that role with other kind of environmental duties potentially. And so tribal staff, tribal organizations are great needing to do that. But like as Shannon said, they're overwhelmed too, and their funds only go so far. So those are all things we need to talk about, and it is a food sovereignty issue. I mean, Karen Pletnikoff from Aleutian Purple Off Island Association, she told me this once, and it just stuck with me, that she says we don't have enough Aleuts left for us to like roll the dice every time we eat our subsistence foods, and that's kind of chilling. And so it's definitely something we have to do. How we do it is going to be the real question, and so you can put in place programs that are called harvest and hold, where you harvest your bucket of clams, you keep it in the fridge, and you send out a sample to the lab. Again, if you're willing to wait 3-4 days at best for that to come back, but eventually, if you want this to be done at a larger scale, the CTOR model is a fantastic model. And it just takes time to set up, it takes money to set up, and it takes a lot of agencies to kind of work together along with the tribal government to get all that set up. So hopefully we'll get there, but it just takes time. But I'm really grateful for CTOR and Sitka Tribe to kind of show the way and lead the way on how that can be done in a very, you said grassroots, but I think it's just a local based initiative. It's not top down at all. It's from the local, whether it's government or not, it's still very locally based, and that's the important part.
Yes. That's all great. It's I have been struggling with this for many years. It is a really hard issue. And I'm working more in the northern, the Beaufort, Chukchi, Bering. There's more resources in the Gulf for that. But let's just recap, for 12,000 years, people have been harvesting whatever they want, however they want, with healthy food, with a specific type of ice cover, certain conditions, etc. I loved, was it Corey Erickson that put that circle up there, and then it was like that 3-year project, and I was like, oh, that's so amazing. You just think about that circle being 12,000 years, and the last 20 years or the last 10 years on there. So traditional knowledge is not equipped for that kind of rapid change necessarily. There's a lot there. There's a lot of information, absolutely, and that these toxins have been there before, but not like this. So I'm just really struggling. Because the consequence is so high. The consequence is death. And in other parts of the world, there are, this is you guys are doing the receptor binding assay, which is a phenomenal assay. It uses receptors, so you're actually getting a measure of toxicity, but it's not AOAC approved for
reopening, and I think it's approved for closures. If you check on muscles,
It's approved to close it, but it's basic. All I'm getting at is the health agencies in the lower 48 or in the more populated areas have worked on this for 100 years or 60 years, and it started with a mouse bioassay. They took extracts and they gave it to mice, and at a certain point, the mice had problems, and okay, this is dangerous for mammals, So that's there. This is where we are, a coastline that is way too big to monitor regularly. And so it's a scary thing. I'm just going to say what I've been thinking is there need to be locally managed programs. And I do want to call out Norton Sound Health Corporation. They're starting, but again, still we're talking Nome. If you have a lab in Nome, which is what I'm thinking, I think maybe the Nome Hospital would be a great place for a seafood safety lab that is up to the entire standards of the DEC, that's operated with HPLC, suite of toxins, regular monitoring, a way for the communities to get their stuff there and back, et cetera, and be told. But that's still, how long does it take to get to Gamble and Savunga from there? So then, so anyway, so that is the issue, but there is progress, there are people that are coordinating. It's gonna have to be in those communities and led by those communities, but it needs support. I believe that it needs support and training and equipment and expertise, and I believe that there's a desire. So that's we're not there, but there's something to look at.
There's a question.
Yeah, can I just have one quick thing? One quick thing is because I was just in that session about fish in the future, and we talked a lot about methylmercury and all that. Yeah, Andrew was there, and the messaging around that, and it is very difficult. And I want to acknowledge that, I said something that was a little scary. Kathy just said something that was a little scary. That this is the consequence is death. Yes, very high consequences. Overall, it is, just to put in context, we've had 6 recorded deaths due to PSP since '93. So that's what we're talking about in terms of number of deaths. Hundreds of over 100 cases in that time frame. So high consequence, kind of low probability. And the point is not to dissuade people from eating their subsistence foods. And so we need to strike that balance when we message and talk about this, just like we were talking about with methylmercury and fish, that balance between informing and not alarming. And that's a difficult kind of balance to strike, but I think we need to talk about it because it's an important part.
Let me say one more thing, because actually that's one thing that always bothers me is the alarmist thing. Yeah. And when I was saying that, I didn't mean it's a big risk we're always worried about.
I know you were.
The point was that saying it's safe. It's when can you say it's safe? That's what they want to know. When is it safe? That's what we all want to know. When is it safe? And that's the answer that is so difficult to figure that out. But we are getting some answers. For example, the communities that hunt walrus and bowhead, we are getting real answers that can inform the harvest. So we're doing a study in our EcoHab project where we're looking. I don't know if we already mentioned that here. We're looking at the different tissues in the walrus, et cetera. So we'll be able to have those answers, and we can comfortably say, even at this point, that mussel and blobber are never going to have levels above or regulatory limits for many different reasons. Chemistry, the animal would die first, et cetera. But there's the topic of the health of the animal and the resource. So that's also a food security issue. How are these blooms going to affect that? So yes, but thank you for the alarmist thing. We don't want to be alarmist, but the consequence is so high for a unexpected bloom, and so it's a really, really hard situation.
I just wanted to emphasize Alaska in this part. Like, Alaska does have so many of these remote communities that are difficult to get to, that can't be reached without boat or plane, and with so much coastline, and with so much reliance and lack of maybe respirators at medical facilities or fully equipped medical facilities. And it is the only state that I think has had a death from PSTs since the 1950s. So 6 is not statistically large for the number of people who consume, but we don't want any, of course. And when we compare it to the rest of the United States that hasn't seen these numbers because they have effective monitoring programs, it really shows that monitoring does work, and that we need to be protecting our resources and protecting our people.
Hello. I was wondering if there's any kind of state or regional surveillance system that connects animal, human, and environmental cases of HABs.
Well, that's kind of what Warren West is. So we're doing surveillance for the presence of toxins in stranded animals and subsistence harvested. There's not a regular. Well, I'm just going to say this. I know there are small efforts here and there, but in general, there is not a coast-wide surveillance of harmful algal blooms in Alaska. So we don't always know when the cells are there. This project that I've been talking about is a summer season of sampling, so we're getting a lot more information, and we can start to make predictions on when we think blooms will be there, et cetera. But so as far as in this way, we are doing that surveillance as a research project.
And just an interesting point for anyone who knows, or even if you don't know, certain blooms you can map with satellite, so you can use that as your surveillance system. But Alexandrium toxicity is high enough at low concentrations. I think it's 500 cells per liter is a concerning level. You wouldn't be able to see that via satellite. And also living in Southeast Alaska. It's gray, it's rainy, rainy. So you're trying to take a picture of the ocean and you just have a photo of clouds. So I thought that was something really interesting because that kind of surveillance hadn't even come to mind for me. But hearing about how it's ineffective for our particular uses at this point in time is just another obstacle we need to work around.
Yeah, there are techniques that are being used. There's a lot of techniques developed for very nice visible HABs that you can just see with the naked eye, and especially freshwater HABs that are in that situation. But there are hyperspectral cameras that are being developed and tested as to whether or not they can detect things. Then there's I showed a picture of one on this corner here, where there was the IFCB. It's the image flow cytobot, which is a little basically a pump that pumps water over a camera that takes a picture of every single cell. And then an AI can actually detect Alexandrium out of that. So you kind of have this automatic detection. They're very expensive. They take a lot of training to learn how to make. California has deployed 10 of them across their coastline. They use that to forecast blooms. So they've got, but that again took a lot of money and years to set that up. And that's California, which is, it's a big state, but it's nothing like what we have. So where will we put 100 IFCBs? I mean, it's just, when you start talking about the scale of Alaska, a coastwise surveillance is just going to be very difficult. You could put one in front of every community potentially, but then you're back to, you'll see Alexandrium quite often in the water. Are we shutting down the beaches every time we see, a certain number of them? They may not be toxic. You may be keeping people away from their resources unnecessarily. It really comes down to you need to test for the toxin at this point, at least.
And I also, for those who might not know, HABs can also be super localized. So we've tested beaches in the same community where the blue mussels are hot or above the regulatory limit, and down the road they're fine. So that's why in our advisories we always list where the species that we've tested are affected. But that's an important thing to remember too. So with deployment or testing or what have you, it's not necessarily representative, and you have so many little bays and coves and outlets all over the coast. Alaska is not just a straight down coastline. You're going to have a lot of different events happening individually.
I don't know, I feel the need to restate that, you had just said it though, that the toxin testing is really going to be important for the toxin testing of the food sources, because you're talking about surveillance on our cruises. We are taking benthic samples, so we're getting cyst counts in the sediments, but we're also getting clams, worms in the benthos. And in 2019, which is the warmest year on record in the Bering Sea, there were 3 locations where clams were above seafood safety regulatory limits. One up near Utqiagvik, one in Chukchi, and one in North Bering Sea, from the bottom of deep water. So that is that. Then there are traditional practices, for example, of consuming clams out of the guts of walruses. And so those walruses are harvesting those clams. So I guess what's difficult, what comes together and is not a clear answer to, is the state has made their statement. Recreationally harvested seafood is not safe to eat, necessarily. We can't say that it's safe to eat. Commercially available shellfish is safe to eat. But there's always a risk of PSP for recreationally harvested shellfish. That's not subsistence harvested. That's assuming that people are recreationally harvesting. They have a choice. They're going to, okay, well, we're not going to eat that. But there's nothing around the subsistence part. And subsistence isn't a choice. So there's no guidance there. So some of these practices, we just want to share the information and figure out how we can test to make sure that seafood is safe. Because eating the clams out of the guts of walruses carries, that's what the state's going to say, carries the same risk as harvesting from a beach or anywhere.
I had one prior question, but now I have another one. The first one is, how long does it take for something for an area to get hot, for example? Because sort of what I envision the future of this is that it kind of comes out like the avalanche forecasts, you know, where everyone knows there's some risks to backcountry skiing, but that they get notified of the relative, what the model is saying, what we know. But then it would be great if people could do something like kind of like the community monitoring that we're doing now, where they have either they know where there's, you know, they can just go pick something off 3 days before they know it's going to be low tide, wade out there, grab something, or, you know, a mussel, because then clams might not be able to access, and send that off. And everybody has access to the U.S. Postal Service. If you guys have shipped anything in Alaska, samples, which I do all the time, you can't be 3 days. It's like they need a week. At least, maybe more. So I'm wondering if it's feasible, really, with the, like, how fast is it going to get hot? And then I already forgot my other question. Maybe I'll remember it.
So I'll start with the first part. I think from what I have seen, it can happen quickly, and that's just for my own testing. But these surveillance almost that we have is communities throughout Southeast are still doing phytoplankton tows. So they're doing those on a weekly basis. And a couple of years ago, Ketchikan actually saw several Alexandrium cells in their phytoplankton tow on their microscopy slide, and they put out a preemptive warning. And I don't think were scheduled to collect shellfish that week, and they did anyway. And they sent it to our lab, and it was really hot. But the previous shipment they had sent, and I remember they were pretty consistent, so that was a week before, 2 weeks before, was fine, or was maybe a little bit higher. So these things can happen quickly, and again, they deprecate at different rates. I don't know how quickly they all take them on, but I have seen blue mussels go from notably under the regulatory limit, maybe 20 or 30, to over 1,000 between 2 weeks. So, you know, I can't say if it's a day or 2 or 3, but I think having that first pillar of warning, keeping an eye on the water column is helpful. But we've also seen at our own beach, PST levels seem to get higher without us noticing Alexandrium in the water column. So both facets of monitoring are really important. And like Thomas said, and Kathy reiterated, I will reiterate one more time, security is in testing. And the great thing about the CEDAR program is that people are able to send community members. There is no testing fee for shellfish shipments. We have our own contracts with other governments about their shipments of shellfish to us, but if an individual wants to know that they harvested this shellfish, is it safe? For PSTs, we don't do any other testing currently. We can provide that information. And really that is the most secure way to know about your sample, because we will put out advisories for, 2 weeks after, or for a month after. If it's been high, it's continued to be listed just for caution. But if you test your blue mussels and then a week later go out and harvest, there could have been a bloom, and if there was a bloom the day after you tested, chances are the blue mussels still have sax toxin. It can pick up pretty quick.
So I'm also in Southeast Alaska, and I'm just wondering what, you mentioned putting out PSAs. What are your best communication tools for just spreading the word for people that maybe don't know to go to the CTOR website?
It depends on what kind of numbers we're looking at. So our partnerships with the other tribes, we leave a lot of that jurisdiction for how they want to handle it to them. So the reports go out to anyone on our listserv, which would be anyone anywhere in the world who wants to be emailed about it. That say the level of toxin, and then the PSA says which beaches are closed for which species, or which beaches are advised not to be harvested to based on which species, because we're not regulatory. But for our own beaches, if we are above, if we are notably above, so that would probably look about 200. I don't remember off the top of my head what our protocol is, but radio contact, Facebook page, Southeast loves Facebook, and word of mouth. You know, Sitka is pretty large compared to some other communities in Southeast, but still you run into at least 4 people at the grocery store. So I think we rely kind of heavily on that percolation of information, and a lot of people are engaged with Sitka tribe and usually do keep up to date and call. People who are unaware of the services that we provide or that information, I don't really know how to reach them. So if anyone has a suggestion, that would be great. But our partner tribes all have their own mechanisms of spreading that information, whether it's a bulletin board or radio or what have you. And for individuals who submit samples, they get an email or a phone call with their results.
Yeah, for at least some of the communities, they have the clam hub billboards right on the beaches
where they're being tested. So I know Sitka does, and there's other communities that have that too. So as you're going down to collect, there's like a sign, like a, you know, bears have been here recently sign, and a, you know, don't harvest the shellfish sign, or you know, risk is high kind of thing. So.
Thanks. And and any communication, so mainly just communicating with the tribes, not necessarily with the health organizations, or would the tribes then communicate with?
Yeah, so the tribes are kind of in charge of how they want to data disseminate. And again, our focus, even though we are a laboratory, is food sovereignty and safety. And it never really crossed my mind to be in touch with the health department because I guess they would catch wind of it if it happened. We've heard of suspected PSP cases, phone calls to our lab, because that's something that's happening at a hospital or at a place where they're attempting to rehabilitate somebody, and they're following up with us about testing. But the individuals in the governments have communicated, and you know, DEC has been wonderful in providing information and helping train, but historically they haven't worked so well with monitoring together. Like that bridge is not built, so I think that communication just isn't a priority, because as long as the communities know that their food is safe and secure and that's getting out to the individuals, that's the priority.
I just had a small suggestion, maybe using just traditional means like Facebook and radio. When I lived in Seward, I know there was tons of salty old people that were like, oh, I've been doing this for 30 years and I don't need to even think about that. And I was constantly telling people, well, it's kind of different now than it used to be, and it's if anybody gets sick directly after you, of course they won't listen to you. So if anyone gets sick, like, this is what you need to do, like, because they would, go off on boats, and that was about the best I could ever do. But I think for some of those folks, just, that's what gets to them is TV and radio, like, just hearing over and over, like, they're, I think people probably got the message that the state says that it might not be safe, but they don't care about that. But if they got more detail, like, oh, you can get your stuff tested, that kind of thing, I think that would be helpful for some of those folks.
I'm not sure how other communities know about accessibility of testing, and even within Sitka, but we do the radio and community information for above, but we also have outreach programs. So there's a lot of like education programs. It's particularly geared towards school age, but hopefully that is transpiring back home about food safety, and like we have herring camp and clam camp, and these a lot of different topics get covered, but there is outreach, and I don't do that, but to make sure that people know what issues are present, and then hopefully they know about the resources available. I also had a thought, I might have spoken out of turn, that the Department of Health, I think, is listed on our listserv. I've never personally contacted them, but I feel like I have seen that they are getting updates, but I can't confirm.
I just have one last comment on that. I think what you're talking about, I do think that there should be a concerted effort in the state of Alaska to train the hospitals and health staff because it's not a common thing, and because you're absolutely reducing the risk by this monitoring, but unfortunately you can never say things are safe to eat. So that communication with the care facilities, I think it's really important to maybe just to re-up that. We had a seminar, like I told you, out at the Nome Hospital, and that was really helpful for that.
I would just like to give a little quick story about a suspected PSP case that happened. They weren't able to get samples from the individual, so at first they called our laboratory and said, we have suspected PSP case, can we send biological samples for testing, which wound up not happening. But there was, from the symptom description and the description of the events that took place, it sounded very much like a PSP case, and the Department of Health was definitely more wary than to label it that way. So again, if people are having symptoms and not going to hospitals, or not maybe saying buzzer words that lead to the identification of PSP, a lot of these cases aren't being documented as PSP. But it's also difficult because we found out where this individual had potentially gotten their shellfish from, with very limited information, and to get out there, it was already 3 days later, and it would take another 2 or 3 days of organizing, and someone else would have to take a boat from a different community. Sitka couldn't do it because it was far away. So I think this program that I've been able to become a part of is incredible, but it's still pretty young. It started in 2014, I think, and testing started in 2016. And it's so multifaceted that it's growing and evolving, but it's hard to sometimes just pack it up nicely, I guess, is a way to put it, because it would have been great if someone's in the hospital to be able to test the shellfish in that area, but it didn't work out.
Does anyone have a last minute question, or do any of our panelists want to make a last comment? Thomas will.
Well, I don't have to be the only one, but if there's no questions, I'll just say that there is like a path forward for increasing our knowledge, increasing our information. We're doing it. Everybody in the network with the research work that's being done by Kathy and all the other researchers is amazing. The on the ground monitoring and information communication that groups like Shannon's is doing is really amazing, and we're getting more and more people kind of sampling and labs hopefully starting up. Nome would be a great one. Unalaska would be another great one. And I think we just kind of touched this point that it's never going to be 100%, but it doesn't mean that we should give up, of course, and we are going to keep working on this threat that is real, and the biggest, I think, tool that we have is communication and outreach, both in people knowing what is out there and having that nuanced messaging about the risk and putting it into proper context, then also that communication after an event or during an event, knowing right away that if you think that you got PSP, you tell people so that people around you kind of can act on that knowledge. And those communication networks need to be local. They need to be kind of locally driven, and that's really where the power of Alaska. We have a lot of challenges, but we have a lot of power, and the power is in our people, in our communities, and so that's who we really need to kind of engage, and a lot of them are there, and we just need to keep working at it. But I wanted to end on a hopeful note, at least for what I'm saying.
That was great.
I think that was well said.
I'll just thank everyone for your interest and for your wonderful questions, and thank you to our panel for providing their expertise. And if you have more questions, please find these guys later this week.
I work for Marine Science with